All right, so in this video, we're going to look at the evidences for evolution. We're going to look at four main things. Okay, so let's just jump right in. Big questions we want to answer. What are the four main evidences for evolution? And these are not specific evidences, all right? These are branches of scientific inquiry that we use to support the theory of evolution. We want to make sure we can identify two different ways we can determine the age of a fossil. We want to know what comparative embryology is. We want to know what homologous structures are and vestigial organs are. And we want to know how biochemistry is used to determine the evolutionary relationship between organisms. So those are the big takeaways from today. So let's start with a simple question. What is evolution? The general description for evolution that most people give is, it's just change in a species over time. We're going to examine that on a little bit closer of a definition, a little more accurate, and that is that evolution is genetic change over time. Now, another way that we talk about evolution is descent with modification, or the formation of new species from accumulated changes. Descent with modification is a really common way we describe the changes that happen in a species from generation to generation, which leads to evolutionary changes. So what are the main evidences? Well, the first one is the fossil record. All right. So when we look at extinctions, we look at the appearance of new organisms, we look at how organisms in a family have changed over time. The fossil record gives us a snapshot of different branches of the tree of life. The second one is comparative embryology. This is when we look at the embryos of different organisms and we compare how they are developing in the womb or in the egg before they are born. The third branch is the comparative anatomy branch, and this is when we look at the anatomy of organisms after they're born, all right? The structure and function relationships between anatomical structures. So like how the arm is built, like what are the bones in the arm, and then what does the arm actually do? Is the structure of the bone related to the function? And then how does that compare across different species? And the last one is comparative biochemistry. And this is when we actually look at the DNA, we look at amino acid sequences in proteins, and we see how similar two proteins are in two different organisms, or two different genetic strands are in two different organisms. So let's look at the fossil record, all right? Fossils are preserved ancient organisms. We find them in sedimentary rocks. You probably learned a lot about fossils in earth science, right? Something dies, it gets covered over with sediment instead of rotting. It's soft tissues or like kind of decay away, but the bones are replaced by rock, and so we're left with a fossil. Age can be determined two different ways when we look at a fossil. We can do relative dating, in which case we look at the things around the fossil and we try to figure out roughly how old it is. And this is an estimate. And in order to do that, again, we got to pull in a little information from your earth science class. We use what's called the law of superposition. So this law tells us that older rocks are underneath younger rocks. And so if I look at this cross section here at the top, I would say that number 4 is the oldest, and letter A, number 1, is the youngest. And that makes sense. Another way that we can do relative dating is using an index fossil. And an index fossil is a fossil that only lived for usually a brief period of time, and it's kind of like a bookmark. So, if you find see one on this that we might be familiar with. Here we go. One of these guys. You've probably seen that fossil before. Well, if you find that fossil, then the rocks that you're looking at are from the Cambrian period, maybe early Ordovician. All right, it just barely bleeds over the line there. If you were to find, one of these guys right here, well, then we're probably talking about the Jurassic rock. So an index fossil is just a fossil of an organism that lived during a set period of time that we've identified, and we can then use them to help us date the rocks that we find them in. Now, sometimes you need to know an absolute date. And so in order to do that, we use radioactive dating. Not radiocarbon dating. You can use radiocarbon dating, but radioactive dating is just using radioactive isotopes in order to date something. And here's how we do that. We know that radioactive elements break down at a steady rate, at a constant rate. And we know the half-life needed for those elements, those radioactive elements, to break down. And so what is the half-life? Well, it's the amount of time that it takes for half of radioactive material in a sample to decay into something else. This is different for different chemicals, different elements. So for carbon-14, it's about 5,770 years. For something like potassium, which decays into argon, it's like 1.25 billion years. So different elements decay at different rates, and so they're useful for dating different aged things. So carbon-14 dating is good for about 75,000 years. It starts to lose its accuracy between 50 and 75,000. But when we plot half-life, we get this curve, all right? And if you know how much carbon should be in it, and you compare it to how much carbon-14 you find, you can figure out where your sample lies on this curve. And so, if it died today, it should have 100% carbon-14. None of it should have decayed away. But as things get older, they come down the line. Let's say you find something, right here. Well, that tells you it's, roughly 27,000 years old. Now, for really, really old things, we can use potassium-argon dating. So radioactive potassium decays into argon. The half-life for this is 1.251 billion years. So if, something today should have, we'll say 200 grams of potassium-40, and I just measure how much is in there, and I follow where it is on the curve. So if it lands here, all right, well, that thing is 6 billion years old. If I find a fossil with early life in it, I would expect it to be right there, right? Between these two numbers, 4 and 4.5 billion years. We know that the earliest fossil we have is about 3.8 to 4 billion years old, but we know it can't be any older than 4.5 because this would be the age of the Earth. All right, so obviously potassium-argon dating is useful for dating things that are older than the planet. So that's a pretty useful chemical to find. Now, comparative embryology is the next branch we're going to just kind of briefly look at. And it looks at the embryos, the developing fetuses of different species, and examines the relationships between them. And, what we find is that the early stages of development for fish, birds, rabbits, humans, all these different organisms are really similar. It's actually kind of difficult to tell the difference between a human embryo and a rabbit embryo if you don't know what you're looking at. So here's some drawings, all right, and I'll show you some actual pictures here in a minute. But just to compare the two, when you look at the same stage of development, so the same overall time, so like obviously human beings are in the womb for 9 months, a rabbit is not, but if you look at the same, overall, we'll say percentage time, and we compare their embryos, we come up with a very interesting set of relationships. And you can see, it makes sense that fish and salamanders look much more similar to each other than they do to, say, a human, right? Distantly related. But now, rabbits and humans look very similar. We're both mammals, we're both vertebrates, right? We have a lot in common. So it makes sense that the development of closely related species is going to resemble each other, and more distantly related species don't. Here's an interesting one, right? The tortoise and the chicken. So we have a bird, right? And we have a reptile, or an amphibian. Reptiles and birds, similar development, right? They look really similar here and here. Okay, so there's some interesting stuff that can be kind of garnered from that. Here is a Myosaura embryo fossil. Now, you want to talk about rare. That's rare. Why? Why would it be rare to find a fossilized baby dinosaur in an egg? Well, think about it. If you're, a predator, right, back in the dinosaur age, what is the easiest thing in the world to eat other than an egg? I mean, it's full of protein. Snakes eat eggs. Today, lots of organisms will eat the eggs of others. So to find a fossilized embryo, that's pretty rare. But here it is compared to a chicken embryo, and you can see what a striking resemblance that there is. Now, comparative anatomy is the next branch. Comparative anatomy looks at the body parts of an organism after they're developed, so after they're born, and it looks for homologous structures, so body parts that are different between related species but come from the same embryo parts. And they also look for vestigial organs, which are leftover parts from ancestors that don't have either a discernible purpose right away or don't seem to be useful. Now, here's a great example of homology or homologous structures. Penguins and eagles are both birds. They both have, arm buds or wing buds in development. But the wing of a penguin and the wing of an eagle are obviously very different. Penguins have short, tight feathers. They're close together. Eagles have much larger feathers. The wing of a penguin is a little bit heavier than an eagle. It also has a different overall kind of function, right? So penguins, they fly through the water, right? Where eagles are going to fly through the air. So they are obviously related. They're both birds, but you have different structures for different functions, okay? Now here's another very interesting one. This is one of my favorite slides in all of biology. So everything that you see right here, the bat, the sea lion, the chimpanzee, and the human, all of these are mammals. All right, so we're all vertebrates. We're all warm-blooded. We all, have hair. We give birth to live young. We nurse our young, all that good stuff. These are mammals. Look at the similarity in the bone structure of the arms. Even though they have different functions, right? They're adapted to different ways of living and different purposes. The structure of all these arms are based on the genetic code. And there is a very high degree of similarity in the arm bones of mammals. So we have one bone in the upper arm, two bones in the lower arm. You have, the wrist, the hand bones, which are the metacarpals, and then you have the finger bones, which are the phalanges. Well, and it doesn't matter if you look at a bat, right? 1, 2, then we have wrist, hand, fingers. Here's a sea lion. 1, 2, wrist, hand, finger. All right. 1, 2, same thing. Wrist, hand, finger, right? All the way down. And the closer the relationship, the more similar they are. So chimpanzees and humans share a very close ancestor, and their bones are extremely similar. All right. So when we see a similarity in structure and function, that tells us there was probably a common ancestor for those species. So let's look at another common ancestor, birds and dinosaurs. So this is a pretty famous fossil, Archaeopteryx. Archaeopteryx is important because not only the bone structure that it leaves behind, but also you can see the imprints of the feathers that this dinosaur had. And this is definitely a dinosaur. Here's another picture of it. It's a little bit better, right? This is a true color picture where you can see I think it's sandstone that it's in. But when we look at these fossils, we start to see things that are, pretty reminiscent of birds that we see today, like, the curved neck in death, the, kind of splayed open wing structure, right? There it is again. You can definitely see that this thing had some teeth, all right? This is a great view of the hip. And one of the things that scientists look at when they're examining birds and dinosaurs as they compare their hip bone structure. Now, what do we get from that? Well, it tells us that birds and dinosaurs have a common ancestor. And so in our dinosaur branch, there is a common ancestor at some point from which modern birds descended. And that's how we describe that relationship. Okay? Now, the last branch is comparative biochemistry. And in biochem, we're looking at the similarities and differences in DNA and protein sequences in different species. So if you're really closely related, you should have similar DNA and similar protein structure and similar protein amino acid organization. Then compared to more distantly related species. You can base an entire evolutionary chart on phylogeny, which is the comparison of genes. And so what are we comparing? What are we talking about here? Well, think back to when we talked about the transcription and translation of amino acid sequences, like how you build a protein. And when we want to study amino acid sequences, what we do is we look to see if the same protein in different organisms has similar protein structure, right? Similar amino acids. So in this case, we're going to look at hemoglobin. So hemoglobin is the protein in red blood cells that carries oxygen. And we're going to compare it in humans, horses, gorillas, chimpanzees, and zebras. So all mammals. Obviously, the horse and the zebra are probably closer related. And then the gorilla, the chimpanzee, and the humans are all in the primate category. So we would look to see if they are similar. So humans go lysine, glutamine, histine, isoleucine. Well, lysine, glutamine, histine, lysine. So we have one change in the gorilla. All right, so we have one different, right? If we say this is our true, we have one different there. In chimpanzees, we go lysine, glutamine, histine, isoleucine, none. All right. In a zebra, we've got one, two, nope, same, different. So we have three. And one, two, nope, not three. So there we go. So now we're comparing the number of differences. So we would say these two are probably the closest related. That would be the next closest. The three sequences, all right, well, that's a difference, right? That's a pretty significant difference. But we can also say that those two are probably related. Okay, so the more similar the sequence, the closer the common ancestor. For some perspective, all right, here is comparative genomics. So we take the size of the genome, the number of genes it has when we sequence it, and we compare what we're looking at. So it's not like, in this slide, I compared 4 amino acids in a sequence. We're talking about comparing between 20 and 25,000 genes, which have millions of amino acid sequences, when you put them all together. And that's what we're comparing to other species. So it's not just 4 amino acids. We're comparing millions of amino acids. And when we do that, here's kind of what we find. When we compare the human genome with chimpanzees and the bonobos, like a branch of monkeys, we have 98.8% similarity. So there is only a 1.2% difference in our genes and their genes. Now, does that mean we came from chimpanzees? Absolutely not. What it means is that chimps and humans share a common ancestor. So a really common misconception in when we talk about evolution is, humans descended from chimps. Well, no, that's not true. Humans and chimps share a common ancestor. Now, gorillas are 98.4. So between humans and gorillas, we would expect, right, and a common ancestor that's further back, right? So the higher you go on the chains, the older the ancestor is. For orangutans, right, it'd be even further because we share less, right? The orangs. Okay, so that's what we're pulling here. Mice be even further, right? Canines way back, avians way back, right? It's only 65% similarity. So you should now know the 4 evidences for evolution. What are the branches of science that we're studying? You should have 2 ways that we can determine the age of fossils, right? Index fossils or relative dating. And then we have radioactive dating, so relative and radioactive. You should be able to describe what we're comparing in comparative embryology. Make sure you know a definition and an example of a homologous structure. A great example of this would be like wings in 2 different, like a penguin and an eagle, or the arm bones or leg bones of say, mammals. Make sure that you can describe what biochemistry uses to determine their evolutionary relationship. So what are we comparing in biochemistry? If you have any questions, let me know, and I'll see you guys in the next video.